Okay, so this is a fly through of carb digestion and absorption. So we've got different types of carbs. We've got starches, glycogens, and cellulose. Starches and cellulose, these are found in plants, glycogens from meats. Important to bear in mind A-level knowledge. So we've got our starches. Starches consist of amylose and amylopectin. I believe they have like alpha glycosidic bonds, whereas cellulose has beta glycosidic bonds. Important to know that cellulose does not digest because it has beta glycosidic bonds, whereas all the enzymes, for example, you've got alpha amylase and all that stuff, that can't work on cellulose because of the beta glycosidic bonds. For that reason, cellulose is undigested, so we'll be looking at starch and glycogen. Anyway, so we swallowed it, or like it's entered our mouth. It's in our oral cavity now. So this is like, the green is everything to do with carb digestion. Ignore all the red, you shouldn't be able to see it anyway. So this is, for example, our little chain over here. We've got a bit branched. Now, what happens firstly is the salivary, or however you say it, the gland releases alpha amylase. So this acts as a molecular scissor, and it just kind of breaks apart these wonderful glycosidic bonds. However, it's important to bear in mind that it cannot break terminal bonds. So yeah, it just turns it into like a shorter chains kind of thing, the polysaccharides. So you can turn it into like your I don't know, anyway, yeah, it's just make it a bit shorter, to be honest, than like the longer chains. Anyway, we go through the esophagus. I'll zoom out, this is the esophagus. Now we're entering the stomach over here. Now we're into the stomach. So we've gone past first step, now we're going to be looking through the second step. In terms of carbs, it skips the stomach. Carbohydrate digestion doesn't really occur in the stomach. So we flip through there, go through it. Now we're into the small intestines and this massive mess. I'll break it down. So here we've got our little polysaccharide kind of thing. Now what happens is this releases, so this is our polysaccharide. Now these are enteroendocrine cells. These are lining it, lining the duodenum. And just by like, I don't know, the polysaccharide just being there, then it triggers the release of CCK. CCK stands for cholecystokinin, I believe, unless it's cystokinin, somewhere around like that. That's CCK. That gets released from the enteroendocrine cells, and that goes all the way into our pancreas. Now, okay, wow, the video's already 2 minutes long. Anyway, CCK then triggers the release of pancreatic juices from the pancreas. So let's just say this is a green line. Pancreatic juices come out. Now, the important one we're looking at will be alpha amylose. Now, this is the same alpha amylose that we had over here, if you can see it without the glare, from the salivary gland. However, important to bear in mind, this alpha amylose that's secreted in the pancreatic juices from the pancreas is much more potent, so it's going to like work much more effectively. Now this starts getting to work on our polysaccharide, and it breaks down the polysaccharide into all of these different things. So we've got our maltose. Bear in mind, maltose, sucrose, and lactose, these are all like disaccharides. I think alpha dextrin also is a disaccharide as well. Maltotriose is a trisaccharide, I believe. But yeah, that's finishes our fifth step, then going into our sixth step. So these are just existing and stuff, and I don't know, there must be some type of interaction between that and the brush border cell. These are all our different brush border cells, except ignore the red one right now, that's protein digestion. Now the brush border cells, these release enzymes. The enzymes they release are our maltases, our sucrases, lactase, and alpha dextrinase or isomaltase. So our maltase, the enzyme, works on our maltose and our maltotriose. Our sucrase works on sucrose, lactase on lactose, and alpha dextrinase, also known as isomaltase, works on our alpha dextrin. In case you don't know, alpha dextrin, it just looks like this. So if we remove this and remove that, it's just like this molecule here, this like disaccharide. Anyway, that's step 7 finished. Then we're going to go into step 8, if I can find it somewhere. Where is step 8 gone? Where is step 8? I feel like, did I miss step 8 maybe? So we've gone through that. Oh, sorry, that's step 8. Okay, cool. So step 8, so all these different enzymes, they break the, I think the peptide, or whatever they are, not peptidase, lol. These different enzymes, they break down all of these into our monomers. So once again, A-level knowledge. Maltose breaks down into glucose. Maltotriose also does break down into glucose. Then we've got our sucrose into glucose plus fructose, lactose into glucose plus galactose, and alpha dextrin into glucose monomers. Now we've got all this ready. We've got all of our different monomers. Time to look at absorption. So if we go all the way down to here, ignore all the red. except for this red, because SGLT, like all this is important. Now here we are looking at our enterocytes. So this is also lining like the small intestine, I believe. So let's look at this. Let's break it down. First step over here, that's what step 8 is. It's over here. First step is our sodium potassium ATPase pump. Now this is over here. I think that this is called the basolateral membrane from Dr. Johnson's lectures. I am not stabilizing the camera. Anyway, so 3 sodiums get pushed out, 2 potassiums get pushed in. Because we got more sodiums being pushed out, what it happens is simple, like concentration gradient stuff. It reduces the concentration gradient of sodium, or reduces the concentration of sodium inside our enterocytes. So what happens from there is, once again, like I said, simple concentration stuff. There's a reduced concentration in the enterocytes of sodium. So sodium wants to then enter the enterocytes via the sodium glucose transport channel, or SGLT. Now, this whole process is either secondary active transport, also known as co-transport. So we've got our sodium, which wants to enter into our enterocytes. However, from this, we can also have a coupling with glucose and galactose. Bear in mind, we're looking at our different monomers, and we want them to enter into our bloodstream. So our glucose and our galactose, they kind of co-transport with sodium, and they all enter the enterocytes. However, for whatever reason, fructose does not enter via this. So fructose enters via the GLUT5 channel, or glucose transporter 5 channel, and that gets entered into the enterocytes. This, however, is by facilitated diffusion, because it's literally just from high concentration to low concentration. Now we've got our galactose, our glucose, and our fructose. They keep moving down. Now this is our next step. Our galactose, our glucose, and our fructose then want to enter the bloodstream, so they enter via GLUT2, and this is via facilitated diffusion, and then they just go chilling into the bloodstream off to wherever they go.